lymphatic system, also the immune system. They're kind of the same thing. Where this figure shows the different lymphatic vessels all over the body. You can see the lymph nodes, the spleen, tonsils, and thymus are the main lymphatic organs. We're going to start off with some definitions. So an antigen is any molecule that triggers an immune response. So any foreign molecule can trigger an immune response. Unfortunately, sometimes your own body can trigger an immune response. Your own cells can. We'll talk about that later. A pathogen is a bacteria, virus, or other microorganism that can cause disease. So pathogens generally are antigens, and that's good. Actually, if it triggers an immune response, you can kill it before it can make you sick. So first we need to know the functions of the lymphatic system. So we said in talking about the blood vessels that more blood is filtered out of the capillaries than is reabsorbed, and the rest of that gets absorbed by the lymphatic system. So there are lymphatic capillaries around all the blood capillaries, and they absorb the rest of that fluid, then they filter it, and they eventually bring it back to the blood. They also help with absorption of fats. Fats are too big to be absorbed directly into the blood, so they're absorbed by special lymphatic vessels in the small intestine called lacteals, and they're absorbed that way, and eventually they make it into the blood. And then immunity. So a lot of our immune cells hang out in the lymph system, in the lymph nodes, and we'll talk more about them. So we're going to watch this animation on how lymph is formed and how it flows.
The lymphatic system consists of lymphatic vessels, lymph, lymphatic tissue, and lymphatic organs. The vessels of the lymphatic system cover the body in much the same way as blood vessels. Lymphatic vessels are filled with lymph, a clear, colorless fluid similar to plasma, but with a lower protein content. Lymphatic vessels have thin walls and valves to prevent backflow. Vessel walls are formed by a thin layer of epithelial cells. Unlike the cells in veins, which are tightly joined, the cells that form lymphatic vessel walls overlap loosely, allowing gaps to exist between the cells. Lymphatic vessels originate in tissue spaces as microscopic, blind-ended sacs within a bed of blood capillaries. Tissue fluid, which is the fluid left behind after capillary exchange, flows into the vessels through gaps between the cells. Bacteria, lymphocytes, and other cells flow in with the fluid. As the vessels progress on their path toward the heart, they converge to form larger and larger vessels. Periodically, the vessels empty into lymph nodes, where immune cells phagocytize bacteria. The vessels continue to merge, eventually forming still larger lymphatic trunks, which drain major regions of the body. The lymphatic trunks converge to form two collecting ducts, one near the right subclavian vein and one near the left subclavian vein. Lymph joins the bloodstream when the collecting ducts merge into the subclavian veins.
Alright, so as you saw, as the fluid leaves the bloodstream, there are spaces in between these cells of the lymph vessels, and fluid can enter there, but they're like one-way valves, so it can enter but it can't exit. And hopefully it brings any bacteria or other bad things with it into the lymphatic system. And then we need to know the way that the lymph moves through lymphatic vessels. It's kind of similar to the way things move through veins. There's no pump. There's not really pressure there. So the fluid moves very slowly. A lot of times the lymphatic vesicles are like next to an artery, and every time the heart beats, that artery is going to expand a little bit. That's going to push on the lymphatic vessels. And it has valves just like the veins to ensure that the lymph flows just one direction. And just like with veins, we had the skeletal muscle pump and the respiratory pump. Those same mechanisms help the fluid move through the lymphatic vessels. Alright, this figure shows the area drained by the different parts. So the right lymphatic duct drains lymph from the upper right quadrant of the body, so that's the purple area on this figure, and it drains into the right subclavian vein. Then the thoracic duct drains lymph from the rest of the body into the left subclavian vein. So upper right half of the body is drained by the right lymphatic duct, everything else is drained by the thoracic duct, and they merge into the subclavian veins. That's how the lymph is returned to the blood. Alright, so lymphatic tissues, they are patches of specialized tissue throughout the body. They house a variety of lymphocytes or white blood cells. Lymphocytes also reside in mucosa linings, where they're known as mucosa-associated lymphatic tissue, or MALT. We'll see some examples of that in a bit. First, we're going to talk about the thymus, so let's watch this animation on the thymus.
The thymus is a primary lymphatic organ that provides the location for B and T lymphocytes to mature. The thymus, which is located in the mediastinum, is divided into lobules that extend inward from a fibrous outer capsule. Each lobule consists of a dense outer nucleus and a less dense medulla filled with T lymphocytes. Immature T lymphocytes, also called T cells, travel from red bone marrow to the outer cortex of the thymus. Inside the thymus, the cells are protected from antigens in the blood, giving them a chance to divide and mature. The developing T lymphocytes migrate toward the inner medulla. As they do, they encounter other lymphoid cells, such as macrophages and dendritic cells. This process trains the new lymphocytes to distinguish between the cells of its host body and foreign cells. Once the training is complete, the lymphocytes are released into the bloodstream.
Alright, so the thymus helps with the maturation of these lymphocytes. So the lymphocytes are produced in the red bone marrow, then they're sent to the thymus. Once inside the thymus, they encounter macrophages and dendritic cells. This trains them to distinguish between the cells of the host body and foreign cells, because if it attacks the host cells, that's not good. That's called an autoimmune disease. And so cells that will do that are actually destroyed in the thymus. But they also have to be able to recognize foreign cells. Once this training is complete, the lymphocytes are released into the bloodstream and they're ready to go. The thymus is bigger when you're younger. By the time you reach puberty, by about age 14, it starts to shrink and its tissue is replaced with fat and fibrous tissue. Once your immune system is set up, I guess you don't need the thymus anymore. Or maybe that's why your immune system gets weaker as you age. So next we're going to talk about lymph nodes. So the location of lymph nodes, they're all over the body. So for example, there's inguinal lymph nodes in your hip region, there's axillary lymph nodes in your armpit region, cervical lymph nodes in your neck. You don't need to know the names of the different lymph nodes. You'll learn some in lab. And if you look at a lymph node, it's made of reticular tissue, which sort of looks like a filter, and it basically is a filter. So lymph nodes, the lymph is going to pass through, and you can see there are, just in this example, there are 5 vessels bringing fluid into that lymph node, and then there's only one leaving it. So this is going to cause a major slowdown inside that lymph node, and inside that lymph node are lots of macrophages, white blood cells, and anything foreign they will swallow and destroy. So cancer often spreads through the lymphatic system, so we need to know why. During surgery to remove a cancerous breast tumor, do they remove the nearby axillary lymph node? that's because cancer spreads through the lymphatic system. More than 85% of the lymph of the breast enters the axillary lymph nodes, making these the most common route for breast cancer metastasis. So if they do have to remove the breast, they'll generally remove those closest lymph nodes as well, then they'll examine it and see if there's cancer cells in the lymph node to see if it has spread. Alright, next we need to know the tonsils. So the tonsils form sort of a ring around your mouth. So your skin is a pretty good barrier to infection, but there are some holes in it, like your nose and mouth. And so the tonsils protect your mouth. They're full of white blood cells, and so hopefully any foreign things go to your tonsils. Tonsils have these crypts in them that would hopefully trap bad things and then destroy them. Alright, next we need to know the location and function of Peyer's patches. So Peyer's patches are found in the small intestine. They're an example of the MALT, mucosa-associated lymphoid tissue. So most bacteria or things that you swallow would get destroyed in the stomach, where it's very acidic, but some do survive that. And the Peyer's patches contain lots of white blood cells to take care of any pathogens that would enter your small intestine. You can also see in this figure the appendix in blue. So if anything got through to the large intestine, hopefully immune cells from the appendix would be able to take care of it and destroy it. So the basis of lymphatic fluid is primarily from the tissue fluid left behind after capillary exchange. Alright, next we're going to talk about the spleen. So the spleen is located on the left side of your body, kind of behind the stomach, just under the lungs. And if we look at it, there's two kinds of pulp called white pulp and red pulp. A lot of blood flows to the spleen, and really it cleans the blood. And there's a great YouTube video about this called "What Does the Spleen Do?" So we need to know some functions of the spleen. So one is immunity. There are a lot of white blood cells in there, and they'll take care of any pathogens in your blood, hopefully. One is the destruction of old red blood cells. So we talked about how those only live about 120 days. And once they get old and non-functional, then the spleen will destroy them and then recycle the parts. Blood storage. So the spleen has a lot of blood vessels and it can store blood. And then the spleen will contract before a race or something. Your sympathetic nervous system makes it contract and give you some extra blood. The other thing that goes on there is hematopoiesis, blood cell formation. So your first blood cells are formed in the spleen when you're a fetus. Alright, so in which lymphatic organ do T cells mature? And they start out in the red bone marrow, but T cells mature in the thymus. Alright, so now we're going to switch more to the immune system and talk about the three lines of defense. So the first line is external barriers. That would be like your skin and your mucous membranes. Where there's holes in your skin, we have mucous membranes. Hopefully that mucus will trap any pathogens and keep it from making you sick. The second thing we have is called non-specific immunity. It protects basically against anything foreign. We'll talk about some of the specific molecules of that. And then the third type is specific immunity. So this is specific for a certain pathogen, such as the chickenpox virus or a specific bacteria. And we'll talk about it also. So for non-specific immunity, these are some of the things So external barriers, really that's the first line of defense, and if something gets past those, then we use non-specific immunity. So phagocytosis, that's our white blood cells would swallow any bacteria and destroy them. We'll talk about antimicrobial proteins, natural killer cells, inflammation, and fever. So let's watch this on phagocytosis.
Phagocytes are cells that ingest and destroy microorganisms and other small particles. If a pathogen makes its way past the skin or mucous membranes and enters the body, it will immediately be confronted by phagocytes. When a phagocyte encounters a microorganism, it sends out membrane projections called pseudopods, or false feet. The pseudopods envelop the organism, forming a complete sac called the phagosome. The phagosome travels to the interior of the cell and fuses with a lysosome. The lysosome contains digestive enzymes that destroy the microorganism.
Okay, so phagocytes are these cells. Their job is to ingest and destroy microorganisms or other small particles. When a phagocyte encounters a microorganism, it sends out membrane projections called pseudopods. Then those pseudopods are going to envelop this particle, forming a sac called a phagosome. It's basically a piece of the plasma membrane. Then it travels into the cell and it fuses with a lysosome. That lysosome has digestive enzymes. It will destroy that microorganism, as we see here. And then the pieces of that microorganism, they may be recycled or they may be released from the cell. And the types of phagocytes, the two main types in our body, macrophages are kind of fixed, and they congregate in areas where microbial invasion is likely to occur, such as in the lungs. Neutrophils roam the body seeking out bacteria. In this figure you're seeing what's called chemotaxis. So that is a neutrophil in the blood, and it's attracted to a damaged cell by this process of chemotaxis, and it can squeeze through the blood capillaries in this process called chemotaxis, and that's how it gets where it needs to go. Next we need to know about some of the antimicrobial proteins. So first we'll talk about interferons. So when a cell is infected by a virus, that's what's happening to this cell on the left, it will start to produce interferon, which then it releases, and that goes to nearby cells, and it causes those nearby cells to produce proteins that will keep the virus from replicating. So if those nearby cells do get infected, the virus can't replicate itself in them. So interferon doesn't help the infected cell, but it helps the neighboring cells so that they won't be infected by the virus. So that's helpful for defeating viruses. Alright, pause for a question. Which is an important white blood cell that travels throughout the bloodstream seeking out bacteria? And the answer is the neutrophil. Alright, next we need to know how the complement system destroys the bacteria.
More than 20 different proteins called complement circulate in the bloodstream in an inactive form. A bacteria, or antibodies against the bacteria, activate the complement. This triggers a cascade reaction in which one complement protein activates the next. The final 5 proteins are called the membrane attack complex. These proteins embed themselves into the bacteria's plasma membrane in ring-like circles. This action punches holes in the bacteria. Fluid and sodium rush into the bacterium through the openings, causing the bacterium to swell and burst.
Alright, so with complement proteins, there's about 20 of them circulating in the blood. Be grateful you don't have to know all their names. At the end of this cascade, we have what's called a membrane attack complex, and they they work their way into the wall of the bacterial cell and form a hole, and that allows water and sodium to enter, and that causes the bacteria to swell and burst, and then it dies. Next, we need to know the function of natural killer cells. So they are a type of lymphocytes or white blood cells. They roam the body through the blood, and they use several methods to destroy the cells. We're going to watch this.
Of all the immune system soldiers, natural killer cells are the most aggressive. They are lymphocytes with no immunological memory and are part of your innate immune system. Natural killer cells make up about 5 to 15% of the total circulating lymphocyte population. Natural killer cells are called natural killers because they do not need to recognize a specific antigen before releasing their toxins and destroying a viral infected cell.
So that green thing you see is a virus that's about to infect this cell.
Or cancer cell. They target tumor cells and protect against a wide variety of infectious microbes. In several immunodeficiency diseases, including AIDS, natural killer cell function is abnormal, thus allowing the viruses to multiply inside of human cells. Natural killer cells kill on contact. The killer cell binds to its target, aims its weapons, and then delivers a lethal burst of toxins that produces holes in the target cell's membrane and creates events that destroy the cancer cell membrane and cleaves DNA into many pieces in a programmed cell death, that is, apoptosis manner. After releasing its lethal package of chemicals, the natural killer cell can detach from the cancer cell and kill many other circulating abnormal cells. Indeed, research has shown that most of the tumor cells that enter the circulation are destroyed during the first 24 hours, and only a few cells succeed in establishing themselves in distant organs from the primary tumor, which is called metastasis. Natural killer cells may also contribute to immunoregulation by secreting high levels of influential lymphokines. Therefore, research to develop natural products to stimulate the natural killer cell activity, especially in immune-suppressed patients undergoing chemotherapy for cancer, or in any viral disease such as AIDS or bird flu, where immune suppression occurs, is very important in the regulation of aspects of the immune system which affects our overall health. Finally, we come back to the beginning of our knowledge of a natural killer cell. Here lies a natural killer cell in a lawn of sheep red blood cells. These cells are perfect targets for the natural killer cell, possessing no MHC1 receptor. The wrath of natural killer cell is fully vented on the surrounding sheep red blood cells. The cytotoxins completely destroy the sheep red blood cells, leaving clear plaques around the natural killer cell. There is no apoptosis event, just direct cytotoxicity. The natural killer cell is one of the key soldiers in the fight against viral and cancer growth.
Alright, so natural killer cells are pretty cool. They go around looking for any infected cells or cancer cells. There's a lot of research with natural killer cells, and can they help us fight cancer? Alright, next we're going to talk about inflammation.
Tissue injury, whether from trauma or infection, produces inflammation. Inflammation stimulates the body's defense system to begin fighting the infection while instigating measures to contain the pathogen. The inflammatory response also includes processes that clean up and repair damaged tissue. Injured cells secrete chemicals such as histamine that dilate blood vessels in the area. Blood rushes in,
process of inflammation, as you just saw, the injured cells will secrete chemicals like histamine that dilates the blood vessels, causes more blood to come into the area. The more blood brings more leukocytes. It also causes these same chemicals cause these cells in the capillary walls to separate, and that makes it easier for the leukocytes, the white blood cells, to leave. And other plasma proteins such as clotting factors leak out as well. The neutrophils will find any bacteria and swallow them and destroy them. They release chemicals that attract macrophages and more cells to the area to clean up the area. And that's basically the process of inflammation. So we need to know the 4 signs of inflammation, how they occur, and how they help with healing. So the first one is swelling. Swelling results from fluid leaking out of the capillaries. And one thing it does, it compresses the veins in the area, reducing venous drainage. It forces the capillary valves open to promote capillary drainage. This helps because lymphatic capillaries are better at removing bacteria, dead cells, and tissue debris than our blood capillaries. So if less fluid is going into the veins, more is going into the lymphatic system, and that is designed for taking care of bacteria and things. Next one is redness, which results from hyperemia, basically increased blood flow to the area. It brings materials necessary for healing, including oxygen and amino acids. Heat is also from hyperemia, the increased blood flow. Heat will increase the metabolic rate and the rate of tissue repair. Then pain comes from injured nerves or pressure on nerves from the swelling, or stimulation of nerves by bacterial toxins. It lets us know that an injury has occurred and serves as a reminder to rest the area so that it can heal. Alright, next we're going to talk about the role of fever in fighting infection. So fever is beneficial with a caveat, as long as it's not too high. Over 104 is really bad. But a slight fever is actually beneficial because it promotes the activity of interferon, and it can inhibit the reproduction of bacteria and viruses. So that's another one of the non-specific defenses. So a body temperature of 98.6 degrees Fahrenheit is commonly considered normal. However, body temperature varies during the course of a day with different activity levels, even from person to person. And depending on where you live, it varies. You also need to know a temperature taken rectally is usually 1 degree higher than one taken orally, and a temperature taken under the arm is usually 1 degree lower than one taken orally, so it depends on where you take the temperature as well. Alright, which statement about non-specific immunity is true? And the correct answer is C. It is present at birth. So now we need to know the difference between specific and non-specific immunity. So we've just talked about non-specific immunity. Basically, it will look for anything foreign and destroy it. With specific immunity, there's 2 types: cellular, which aims to destroy foreign cells or infected cell hosts. So one of your own cells might be infected by a virus, for example. And then there's humoral or antibody-mediated immunity, and it focuses on pathogens outside of cells. It produces antibodies that are going to mark pathogens for destruction. So we need to compare and contrast active and passive immunity and describe what a vaccine is and what it does. So active immunity is when the body makes its own antibodies or T cells. So this happens when you get sick. You make antibodies or T cells in response to this pathogen and you destroy it. Passive immunity is when immunity comes from you receive antibodies from another person or from an animal. So passive immunity can happen naturally. A fetus acquires antibodies from the mother, or a baby gets antibodies through breastfeeding. You can also receive passive immunity. They might inject antibodies that are produced in an animal, such as for treatment of rabies or botulism. Now people are working on using antibodies from people that have had COVID-19 to treat people that are sick with it. So that's active and passive immunity. Active, you actually make your own antibodies or T cells. Passive is generally you're getting antibodies from another source. So passive immunity is not going to last very long. Active immunity should last a long time, hopefully your whole life. And what a vaccine is, a vaccine generally contains dead or weakened pathogens, so that way your body is able to recognize the pathogen and it can make antibodies to it. But since it's dead or weakened, it doesn't actually make you sick. So it's a way to get active immunity without actually getting sick. Alright. Next we're going to talk about lymphocytes. So T lymphocytes mature in the thymus. Again, they start off in the red bone marrow. B lymphocytes mature in the bone marrow, and then they'll travel to the lymphatic organs and other tissues. We need to know the structure of an antibody. So here's what an antibody looks like. It looks like a letter Y, and on each end there's a binding site, and as you can see, this one binds to this round looking antigen, but we would have other antibodies that are specific for different antigens. So you would have antibodies for chickenpox and antibodies for influenza and antibodies for different bacteria. Next we need to know the 5 classes of antibodies. So IgA, you would find these in the mucous membranes, like in the intestines or the respiratory tract or the urinary tract. These are also found in saliva, tears, and breast milk. IgD, these exist in the blood in very small amounts. They may activate basophils and mast cells, so they're involved in inflammation. IgE are the ones involved in allergic reactions. IgG is the primary antibody of the secondary immune response. It's also the most abundant of all the antibodies, making up 80% of all circulating antibodies. IgM is active in the primary immune response. It's also involved in agglutination of incompatible blood types. So IgM is kind of the first one produced, and then IgG would be produced second. Alright, so cellular immunity destroys pathogens within a cell, and it uses the T cells. The 3 classes are the cytotoxic T cells, or sometimes they're called killer T cells. They actually carry out the attack, so they're going to recognize the infected cells and they're going to release chemicals to destroy them. Helper T cells have a supportive role. They have to activate the cytotoxic T cells. And then the memory T cells, you produce a few of these and they hang around for in case you're reinfected with the same pathogen, and hopefully you'll be able to respond to it quicker and destroy it before it even makes you sick. Alright, let's watch the animation on cellular immunity.
Cellular immunity destroys pathogens that exist within a cell. The immune process begins when a phagocyte, such as a macrophage, reticular cell, B cell, ingests an antigen. The phagocyte displays fragments
Alright, so the immune process begins when a phagocyte, such as a macrophage, ingests an antigen. In this case, it's a bacteria. Then now this phagocyte is called an antigen-presenting cell, so it's going to go to the lymphatic system, probably to a lymph node, and it's going to take those antigens there, and then when the T cell that's specific for that antigen binds to it, it becomes activated or sensitized. Then it starts to divide, and it's going to form clones of itself. Some of these become the cytotoxic T cells, some become the helper T cells, and some become the memory T cells. The cytotoxic T cells are going to actually go around and find this bacteria and destroy it. And the helper T cells support this attack. One thing they do is secrete what's called interleukin, which will attract more neutrophils to the area. And it also stimulates the production of T and B cells. And we need to know why the cellular immune system attacks transplanted organs and tissues. So it's designed to attack any cell that's not your own. So cells have a sort of a molecular fingerprint on them, and your own body cells have a unique fingerprint. So organ transplant is going to have a different fingerprint, and so your immune cells may recognize it as foreign and attack it. So that's why people that get organ transplants have to take immune suppressive drugs, and they have to be careful. So that's what I just said. Alright, next we're going to look at humoral immunity. So this focuses on pathogens outside the cell. This uses antibodies.
Humoral immunity uses B cells that contain thousands of receptors for a specific antigen. When an antigen specific to that receptor comes along, it binds to the B cell. The B cell engulfs the antigen, digests it, and displays some of the antigen's fragments on its surface. The helper T cell binds to the presented antigen and secretes interleukins, which activate the B cell. The B cell begins to reproduce rapidly, creating a family of identical B cells that are programmed against the same antigen. Some of these cloned B cells become effector B cells or memory B cells. Most of them become plasma cells. The plasma cells secrete large numbers of antibodies. Antibodies stop the antigens through a number of different means, including binding to the antigen's attachment points to prevent it from attaching to a human cell, or setting off the complement cascade, which destroys the invading microorganism.
Alright, so we need to know how humoral immunity helps the immune system destroy pathogens outside the cell. So the surface of a B cell has lots of receptors for a specific antigen, and when that antigen comes along, it binds to the B cell. The B cell will engulf that antigen and then digest it and then display some of the antigen fragments on its surface. Then it will find a helper T cell that has a receptor for that same antigen, and when that helper T cell binds to the B cell, it secretes interleukins, which activates the B cell. Now the B cell starts to divide rapidly, so it produces several of these cells, and they're all programmed for that specific antigen. Some of these clone B cells become effector B cells or memory B cells. Most become the plasma cells, and then the plasma cells are going to secrete large numbers of antibodies. And then those antibodies are going to bind to the antigen and stop it. Some ways they work: they can bind to the antigen's attachment points, and that can prevent it from attaching to a human cell. So with coronavirus, they have these spike proteins. If we could get an antigen that would attach to those spike proteins, then it wouldn't be able to get into our cells and infect us. Antibodies also can trigger agglutination or clumping. That helps to contain the antigen, and it makes it easier for the phagocytes to come and do their work to swallow it. It can Antibodies also promote the binding of complement proteins to the invading cell, so activating that complement cascade, which pokes holes in the organism and destroys it that way. Next, we need to know the time difference between exposure to the antigen and the rising level of blood antibodies between the primary and secondary response, and why is the secondary response faster? So if we look at on the left here, we have the primary immune response, and on the right is the secondary immune response. So in the primary immune response, we have to find this antigen, then we have to create B cells, and then they have to make plasma cells, and that takes a little bit of time. So the time between exposure to a new antigen and when we find antibodies in the blood takes anywhere from 3 to 6 days, and so during that time you're getting sick and displaying symptoms probably. But once that primary exposure is over, you keep some memory B cells. So if you're exposed to that same antigen again, you can make antibodies much faster. You can see the secondary immune response is faster and larger. The secondary response takes only hours instead of 3 to 6 days, and so usually it will attack the antigen and destroy it before it has time to produce any symptoms. So this is why like once you get the flu, you generally don't get it again. This is why vaccines work. They allow you to produce memory cells for the specific antigen, and then if you're exposed to it, you get a much larger and faster antibody response, so you can destroy it before it can make you sick. Alright, next is allergic reactions.
An allergy is a condition in which the immune system reacts to environmental substances called allergens. For example, ragweed pollen is an allergen. When someone with a genetic predisposition to an allergy, such as ragweed, is first exposed to the allergen, the body responds by producing large amounts of the antibody immunoglobulin E, or IgE, specific to ragweed. These antibodies bind to mast cells. Mast cells reside in the skin, the lungs and upper airways, and the lining of the stomach. Although this response does not produce an allergic reaction, the person is now sensitized to ragweed. When the person encounters ragweed at a later date, the allergen binds to the antibodies already in the body. If the allergen links two or more antibodies, the mast cells release histamine and other inflammatory chemicals. Histamine causes inflammatory responses that produce the symptoms of an allergy, such as runny nose, watery eyes, congestion, and hives.
Alright, so upon first exposure to an allergy, you produce the IgE antibodies, and now you're sensitized to it. So, a hypersensitivity or allergic reaction is an inappropriate or excessive response of the immune system. So this is when your immune system responds to something in the environment that most people can tolerate. And so when you're exposed to it, you produce the IgE, you're sensitized. If you're exposed to it again, the allergen then can bind to those antibodies, and if they link, the mast cells release histamine. That can cause inflammation, that can cause the runny nose, watery eyes, congestion, even hives. Then the last thing is autoimmune diseases. So these occur when antibodies attack the body's own tissues. The exact cause is unknown. Inheritance plays a role in some of them, and they seem to be more common in women. So some examples of these would be rheumatoid arthritis. So your body might be exposed to an antigen that's similar to your own cells, and then your body starts to make antibodies to that antigen, and if they're close enough to your own cells, then you can start to attack your own cells. Not good diseases to have. So which type of immunity uses T cells to destroy pathogens within a cell? And the answer is cellular immunity. That's T cells. Humoral immunity is B cells. That's using antibodies.